if everyone could come join us. We're next going to have the final two speakers of the symposium and then a discussion. And so our first speaker is Dr. Michael Zasloff, who, as his biography notes, was an MD, PhD graduate from NYU, has served as chief of the human genetics branch at NICHD, and has been involved also in industry, in Magainin Pharmaceuticals, and at Penn. I noticed joining Magainin and Penn in the same year. That's probably a very uncommon accomplishment.
Answer the quota.
Yeah.
How do you do this?
Yes, especially more than 20 years ago. So that's really bringing both a basic science, a clinical, and an industry background to this topic is really helpful. You're currently at Georgetown, right? And he's been including working on studies of innate immunity with the transplant group in the Department of Surgery. So we're really happy to have Dr. Zasloff talk on innate antimicrobial mechanisms in prevention and treatment of disease.
Thank you. I guess this is fine. Thank you, Jesse. And Eileen and David, thank you for the invitation. I'm going to focus this particular presentation, not so much on the microbiome, but on our attempts to perhaps mold those organisms and deal with the organisms that we are constantly in contact with, which in states of health appear as harmony, or as clinicians we would say, a healthy state. Is this? I really didn't want that one. Okay. Anyway, let's talk about, this one, I thought I'd put another slide in there, but this really does make a point that I want to bring up to you, that if you throw away much of your current immunology text, you would be hard-pressed to imagine that this creature could possibly grow to this extent and live. And the only point I want to make is that this animal is surviving quite well on the defenses that I'm going to discuss, some of them. And we have this, but because of the intense focus on disease processes and immunology and vaccination, we have really not paid much attention to this. And I don't know if this has a light organ or not, but if it does, it's a beacon. I got into this somewhat serendipitously. I was at NIH working with, this was about 1,000 years ago, working with Xenopus, and we were doing some studies that required removal of the ovaries. And my early surgical techniques weren't that bad, I thought, and suture this animal, and about a week later this wound heals, and then this wound really heals. And what struck me many years into this work was that this is kind of a miracle. This is not a sterile post-operative suite, and somehow this is clothing, as if it didn't really didn't care. I would have thought that this would be the sort of naive response, but the reality is that this animal is about to die, and you only see inflammation in this creature when it is about to die. And I have the slide. And what happened here was that it turns out that this frog, and actually all frogs, have what are called granular glands, which are juicy nerve endings, that have a dozen or so antimicrobial peptides. And when given the right stimulus, here it's some noradrenaline, they secrete. It could be a reflex, but they secrete. So in the real world, if they get a wound, there's a reflex, or they damage the epithelium in the local area, this gel is laid down that, in a sense, sterilizes, and I would say sterilizes that site during permitting epithelialization to take place, and then everything heals and the system restores. What's interesting, and here's the beginning of our communication, is if you take a frog and you deplete all of its antimicrobial peptides in its skin, you do this by repeatedly administering epinephrine, and then you replace it you put it back into a sterile tank, which has basically been sterilized and maintained with in the presence of where sterility attempted by adding antibiotics to the water. What you discover is that the glands don't restore themselves. The antimicrobial peptides have no stimulus to reappear. In contrast, if you just keep the water normal, keep the ambient bacteria around, everything returns. So this frog has evolved a relationship with its outside world. The molecules in the skin that were present in the skin were antimicrobial, very specific, it turns out, for bacteria versus endothelial cells. And what I'm telling you now is characteristic of essentially all that have been discovered since then. There are thousands from many, in fact, most living creatures, including plants. So a fluorescently tagged molecule will, when put into a medium that contains, or a culture dish that contains an endothelial cell and some Staph aureus, will be attracted to the Staph aureus. The basic design that has appeared throughout nature is very simple. If a molecule can form this type of structure where positively charged amino acids are physically segregated in the folded form from hydrophobic residues, that molecule has amphipathicity and almost always, almost without exception, will exhibit an antibiotic activity. Its spectrum will be different and so on, and these reflect subtleties of structure. George Post will recall something. We had colleagues together. With Smith Klein, we began the development of this. And Jesse, I did go from bench to FDA, advisory panel, and so on. And this is still, believe it or not, in development. So by the time I no longer remember what I've been working on, it will be, I expect, approved. And the FDA really said, wait, we don't want to use the jargon non-inferiority trial. This is working too well. We need a superiority trial. In other words, you have to compare something to a placebo. The treatment group were people who had infected diabetic foot ulcers. Nobody believed that we could actually generate a placebo-controlled trial, but actually it is going to be done. As far as application, as I just described, versions of Magainin have been in trial, and we've published what is a successful phase 3 trial on this. This is a topical use, but the field has expanded because of the simplicity of the target and of the mechanism. It's been possible to generate very simple molecules that can be cheaply made, that work systemically, that are in trials, and this is just one example of a molecule, very simple molecule, that was generated by Bill DeGrotto and Michael Klein at Penn, and it is in trials. A little bit about ecology and this system, the frog. This is a wonderful grandchild experiment or child experiment if you can get your hands on these creatures. These are different frogs from different environments. Here are plates of bacteria, Staph epidermis, E. cloacae, Staph aureus, and so on. And what I show you here is if you take the secretions, the ones that I described as I showed you that we could generate from Xenopus, and you put those secretions unfractionated on a lawn of Staph epidermis. Here we've taken dendrobates and we put it here, and here we've taken Xenopus and we put it here, and so on. What you can see is that the biotype or the range of organisms that these mixtures, there are about 12 or so in each of these animals, can kill different. And the way I look at it is that this creature has evolved in one habitat, and it stays in that niche. It eats food in that niche and so on. It doesn't wander. Xenopus has its own, this particular water frog its own, and so on. It turns out that no two frog species share the same peptides. They're all different. It has been given the opportunity to drift. And as I said, essentially every creature from the single-celled bacteria, of course, up to us, fungi, and so on. Plants use these in many contexts. I'm talking about one class, the class of antibiotic peptide that targets a membrane, damages the membrane, but there are others that have to be considered within this arsenal. Lactoferrin and Lipocalin Psoriasin, Paul Fidel talked about a molecule that is essentially Calprotectin. These chelate metals and tend to reduce the nutritional environment that many bacteria require. And there are lectins like this reg 3 gamma that kills, however. And then there are a whole set of proteins and peptides that act very much like the antimicrobial peptides. They're strongly basic and damage membrane. And we've seen a slide like this. All I would like to say is that anywhere on the body where a bacterium, an organism, fungal organism, could creep in from the outside, we find an array of antimicrobial peptides. They're different. How they're regulated is different. What stimulates their expression is different. Their constitutive levels are different, and so on. And to some extent, I just would like to make the point that they help shape these microbiota. So these are interplaying, and I'll show you in a moment. Here's a paper we published on this subject in a long time. That's not 1,000 years ago, but some long time ago in Science. And it deals with this complicated issue. As a doctor and father, I do not believe that this could possibly remain without inflammation. I just can't see it. I don't care what you tell me about regulatory T cells or anything else. I just don't believe, but it's true. So how does a solid wound like that work? And we decided that this would be an easier subject to work. These are cows. If you isolate, which we did, the antimicrobial activity in a cow, in the cow epithelium, and this in fact reflects the discovery of the first of the epithelial defenses. You discover when we stain with a particular stain that the protein is distributed in the intact epithelium of the cow right on the top, in the top layers. And you see how thick the layer is. The reproductive or germinative layer is down here. When you injure that animal, within a few days, within about a day or so, the wound is covered with an increased expression of this antimicrobial peptide and others. So what's happening is that this wound is gaining yet a greater level of protection. So I want you to appreciate that. There's a constitutive level that probably plays a role in some degree of maintaining the flora. Things that can't deal with that are not going to be happy. Things that can will grow, etc. Once you have an injury, you change the flora locally. What's interesting is that if you sample the cow at birth, it has nothing on that tongue. But if you wait to the point where it has weaned, it is already, and this is almost certainly induction. This is induction, I suspect, by, again, commensals that it is acquired. These are made by animals, and they, like everything else, are part of a system. This very widespread molecule that's present in the human speaks to a receptor, several receptors that then stimulate angiogenesis. What they do is basically, once their expression is raised, there's a wound, there's some reason for their being around, and then they participate in the repair process. And that's really what I want to mention. So some of the pharmacologic uses, and these have been being played with, of compounds of this sort, focus not on the antimicrobial activity, but on the other reparative effects that they have, pharmacologic effects they have in animals. We talked about skin, and I'm going to take you to a different direction. I have great difficulty as a naive person understanding why a well with all this wonderful fluid and moisture doesn't look like this all the time. Bacteria have no difficulty, in principle, going down here, moving down. This is like the best incubator you could ever dream of. It doesn't happen. It happens when something goes wrong with either bacteria or hosts. This entire system is highly supported by antimicrobial peptides at different places throughout the gland. And this, appreciate, is as complex an ecosystem of the human body, I'm sure, and is to be studied. Now, I'm going to show you something that really will help those in the audience who do not want to use ethanol or soap again. You'll be impressed. This was a wonderful paper that appeared some years ago from the group in Kiel, and they discovered this protein present on the skin. It's a relatively small protein, and it's present in higher amounts, greater amounts, nanograms per square centimeter. And you see a lot on the head, you see a lot on the hands, you see a lot in the back and soles of the foot. So there's a relationship between the density of organisms or certain organisms and the expression of this stuff. And it's made throughout the various structures of the skin and other glands. And you should appreciate that this operates by chelating zinc. Well, here's an experiment that it was published in that paper. If you take a lawn of E. coli, now just E. coli, and you put it on your hand, and then you plate it on a fresh LD agar plate. You hold your hand up for 5 seconds, this is what you'll see. But if you hold your hands for 10 minutes, it's gone. And it's gone because of the action of psoriasis. And it's been shown very clearly by inactivating this effect of an antibody. So now, this speaks to the ability of our bodies to mold what it is that, or influence what it is that will land or take root on. So it is maybe not surprising that if you take some soap and you wash your hands, you remove some of those. And at least as far as E. coli goes, it no longer can be killed at the same rate. So hand washing, which is good in certain contexts, is not good, and this perhaps was, speaking to the excessive use of skin cleansing. In the mouse, one particular antimicrobial peptide expressed in the skin, when knocked out, will make that animal, which is susceptible to streptococcal skin infection. And this is shown, this was shown some years ago using a mouse that was knocked out with, had this particular gene knocked out. The point I want to make again is that we constitutively generate antimicrobial peptides that help mold and influence what type of organisms can live on us. A little bit of the skin, and this is an area that most dermatologists don't appreciate, but appreciate this now. This is the skin. It's got a dead layer. It's got a juicy proliferative layer. And the cells over here spit out little structures called laminar bodies that contain lipids and contain antimicrobial peptides. And most of the antimicrobial peptides of the skin are generated in this way. And this layer is sort of like, think of it as bricks. They're dead, and they're bricks. And in between the bricks, there's mortar. And these little lamellar bodies just fill that space. So what happens is the skin coordinates the generation of a water barrier as well as an antimicrobial barrier. And this is created in sort of harmony or together. This is remarkable. We're talking about application, but here's some remarkable biology that is completely non-intuitive. This is done in the mouse, but it's true in man. This is an antibody. This is an antibody to this cramp or LL37, this antimicrobial peptide. If you tape strip, take some Scotch tape, and you do 2 or 3 of these procedures on a mouse. You can do it on yourself. 4 hours later, you see an enhancement of this barrier. In fact, if you look at the RNA, levels of RNA expression in the lower regions, you'll see that a signal has already been transmitted. to the basal keratinocytes. We got a problem, and they're making more of this stuff. If you wait 4 hours, as I said, you fill up. Now, if you put an occlusive dressing on the skin, some tape, some saran wrap, this doesn't happen. It needs a drying effect. So it leads us, those who care for wounds, have to think a little bit now, again, about this sort of thing. And it's not trivial, because this is a person who has diabetes. And this is a person who had some Scotch tape. She was trying to get rid of a duct tape of a callus, and she pulled it off, a tape strip. And this is what happened. So the point I'm making is that something to do with this process is probably involved in one way or another with what is going on in the diabetic. Now, this won Finson a Nobel Prize. This experiment and this procedure was discovered turn of the century, last century, and it started the whole phototherapy movement to tuberculosis. There is no question that this works. It took some time, however, to understand it. And here is the way it currently is understood. You and I may get a bad sunburn. We've definitely had it all our lives. It is almost unheard of to see infection. I don't know of any. It's extremely unlikely. It appears, and this we know, what occurs in humans is that when sunlight converts 7-dehydrocholesterol to D3 in the skin, which then goes to, is hydroxylated, then further hydroxylated, interacts with a receptor, a vitamin D receptor, activates LL37 gene. This has a vitamin D receptor on it, which requires activation. This then leads to the production of LL37 in the skin. And this is a very simple experiment, another application. If you take a vitamin D analog, very potent chemical analog, and put it on the skin, and a few hours later assay the skin, or a day later, you will see induction. So we can induce the levels of this antibiotic on skin. This has a systemic effect. This is where skin and the innate system internally operate. D3 is normally converted to 25D3 by the liver, and then 25D3 goes to all sorts of places. It also goes to a number of monocytes or macrophages, and if that macrophage has through a particular TLR, liganded, grabbed onto a fragment of a proteoglycan, let's say from Mycobacterium tuberculosis, it will, I think I have this, and that's a crazy thing, it will induce, what is induced is the next stage in the hydroxylation of this vitamin D3 precursor, which then activates LL37. And it is very easy to show, it has been shown in this paper in Science, that this will lead to the enhanced killing of tuberculosis. LL37 is quite active against Mycobacterium tuberculosis. The mouse, by the way, does not have a vitamin D receptor on LL37, so the mouse does not care about sunlight. And this whole system is irrelevant for the mouse. I just was going to make a point, associations we don't like, but just appreciate that there are many reasons to explain the weather cycle, the seasonal cycle of influenza. Appreciate that LL37 has been shown to be active against influenza, and also appreciate that in most populations there is a seasonal variation in systemic vitamin D levels. I'm going to spend just 2 minutes on something. These are conditions that many of you have seen. This is a little child with eczema. This is an individual with psoriasis. What is amazing is that there are two systems where the skin is broken. Skin is permeability barrier is shot. This one becomes super-infected. This one never becomes infected. And this was an observation made by Christopher's some years ago. In eczema, let's see where eczema is, chronic dermatitis, atopic dermatitis, if you look for LL37, what you discover is this is a person with eczema. You see a marked reduction, and we'll just look at LL37. And it appears that the presence of a number of the allergic cytokines have interfered with the expression of this antimicrobial peptide. That's the assumption. That is the explanation for what I've just described. In psoriasis, as Yasmin just referred to, the story is completely different, in large part because there is this massive expansion of IL-17 exposure. There's an enormous exposure of IL-17 to the skin, and if you scrape those scales off, one can actually isolate antimicrobial peptides, and this is how the first of the human antimicrobial peptides were isolated. Just to show you another use, some years ago, working with a plant company, we were able to show that if you took a poinsettia and you put in an unregulated fashion one of the frog peptides and exposed these cultivars to a smut, a fungus, this unregulated peptide, present in very small amounts, was able to provide some level of protection, which gives you a sense of probably why these molecules have been so widely distributed. I'm thinking not to talk about cystic fibrosis. I want to go to something else. I want to go to this. And George, don't call this entertainment, okay? It is entertainment, but it's meant to be provocative, because I have no answer to this. Now, I think if one of us were bitten by a shark and dangled our foot in the ocean, we would not be happy. We would almost certainly die of sepsis. The shark has all sorts of vibrio on its mouth, and these are amongst the most difficult infections to treat in man. Well, what's crazy is that not really appreciated is the fact that the dolphin, who lives with a mammal like us, with an immune system essentially like us, if you study it from a systemic perspective, will heal, and this is a bite like this. This is the mouth of a great white, without infection or inflammation in the ocean. It has virtually no blood loss. It has virtually no regeneration. It regenerates this wound. It does not heal by the way we heal. It actually will regenerate this complex tissue, leaving virtually no scar, and there is no evidence of pain. Okay? So this is crazy. This is an animal, 7 days, perfectly, untouched with respect to behavior. The blubber of the animal, which is a complicated composite, starts to cover the wound. It eventually debrides. You see what is granulation tissue, but the granulation tissue is growing up from the base. And then when everything has reached the surface, it closes. And you'll see it close, and eventually there will be no scar. But this is 40 days, no antibiotics, nothing. No suturing, nothing. And it's living in the presence of microbes. Okay? Well, it turns out that if you look at, and this has to be validated, so I want you to appreciate that this still, in my opinion, is mysterious. But if one searches the blubber, if you look at the blubber, and this has been done by others, not me, what you find in that blubber are organohalogens. And these organohalogens are very similar to the organohalogens, the antibiotics, that are produced by the plant life and the animal life, and probably bacteria that live in the ocean. These are the substances that these creatures have evolved to survive the universe, or to deal with the universe of microbes in their world. And this animal, I believe, has retained them, and it's retained them in its blubber, and it's selected from this array, this vast genome of creatures living in this world, those that are effective and are compatible with its physiology. So another way of isolating antimicrobial agents would be to study not necessarily the microbes, but animals like this one. And just to show you, this is almost impossible to imagine to be true. You can't imagine that a wound of this type. This is an animal that never touched land. This one was seen off of a boat, healed like this. And I say that this is probably the most perfect example. This animal has achieved, as a mammal, warm-blooded mammal, the most perfect solution to the existence, its coexistence with microbes. So thank you, and that is the final message.
Thanks very much, Dr. Zasloff. Questions? Okay. Well, it is. I've noticed in marine mammals in general that they often have in fact, people recognize them by the whales, by these scars that in a person would be quite formidable. So that's really interesting. Okay, well, we do have an opportunity for further discussion after the final presentation.